DNA replication. So this is chapter 12. We're going to do DNA replication, and I'm going to write on this slide because I like the way the yellow looks. But we're going to do DNA replication. The mechanism of recombination, which is the mechanism of crossing over, is a little complicated. So we're going to focus on DNA replication, and that's just sort of one of the main parts of the central dogma of molecular biology. So DNA, RNA, protein, and we're doing this part. And so that's DNA replication. This is transcription, and we abbreviate it TXN. This is translation. We're going to get to all these things. The trying to see this on PowerPoint, their figures is pretty difficult. So for the most part, maybe I'll point out a couple things on their figures, but I'll mostly draw it out because I think it's easier to understand. All right, so let's go. And there's also, man, that sounds pretty fast. A little skeptical of 1,000 nucleotides per second, but we'll take their word for it at this point. It has to be very accurate. And it turns out DNA replication does make mistakes, but there are mechanisms for repairing those mistakes, and that keeps the overall error rate relatively low. And so we'll talk a little bit about how some of those mistakes are corrected during replication, and then some of them get fixed after replication, and we'll just talk about that a little bit. So when they were first trying to figure out all these different how DNA was replicated, so Watson and Crick had come up with the structure of DNA. Watson and Crick and Franklin, of course, and so there's lots of different ways that you could possibly replicate the DNA. So if we had two strands, we know that A has to pair with T and G with C, so they knew that part. It's possible that you could just copy both strands and then put the two original strands back together. So if red is our new DNA and blue is our old DNA, that would be the conservative method where you're conserving the original structure. You could have another possible method where you start out with the old DNA and then you make new DNA, and the new DNA is dispersed throughout the new and old DNA are evenly sort of dispersed throughout the new molecules. So that's the dispersive model. But what we know now, and there's a very famous experiment by Matt Messelson, that showed that DNA replication is semi-conservative, where each new double-stranded helix has one old strand and one new strand. So that's a semi-conservative model. I'm not going to go into the details of the Messelson experiments. They're very famous. They're old, and it takes a long time to explain them. They're pretty cool, but I think in the interest of time, we just want to focus on what we know about this stuff and sort of the details of replication. So DNA replication is conservative or semi-conservative. So we have this guy. We got our old DNA is blue. We got two new strands, and when we make two new strands, we got one blue strand and one red strand. Dispersive is just kind of mixed throughout, and conservative, you notice you start with two old strands. Those two old strands go back together. The new strands are all new DNA, and then at each time you have replication, you conserve the old strand, and all the new strand is new. But what we know is it's semi-conservative. So that means you take a double-stranded DNA molecule, you denature it, or somehow separate those two strands, and then make a new strand that's a copy of the old strand. And then these guys are identical to each other, and they're identical to that in terms of DNA sequence. So that's basically what semi-conservative means. There are various modes of replication. Not very many. There's just a couple. The one we're going to be talking about is theta. This is the main replication mechanism, and that was the original that was discovered in E. coli. And then we'll talk about some of the variations or some of the advances on that in eukaryotes. Rolling circle, we're probably not going to talk about that too much. That's another sort of obscure mechanism for replication. It's kind of cool, and maybe we'll talk about it just a little bit. But the main thing is to understand the fundamentals of replication, and then you can apply it to everything else. So here's theta replication, and if there are any sorority women in the audience, the theta structure, the theta Greek letter theta looks sort of like that. And the idea is that at this stage, this looks like a theta. And it doesn't look that much like a theta, but it looks kind of like a theta. And so it's called the theta mechanism of replication. What's happening is we have an origin of replication where we have to start, and then we start to unwind the DNA, and the replication is actually going in this direction and in this direction at the same time. And then eventually both circles get replicated, and then sort of like some kind of magic trick where you separate the two rings, you end up with two copies. This is an electron micrograph, so this is an EM picture. You can actually see DNA replication in process, which is kind of cool. And so this is a replication bubble. So this whole thing is a bubble. And then each part of the bubble is called a fork, replication fork. And one replication fork is kind of working its way around this way, and the other replication fork is working its way around this way. And then it's still going this way and this way, and eventually it gets to the end, and somehow this gets resolved, and you separate into two circles. So we're going to focus in on what's happening at this replication fork and look at some details. And the first thing to keep in mind is that DNA replication has to follow some rules. And so we've got double-stranded DNA, and we always have a 5' end and a 3' end. And so for understanding DNA replication, it's critical to understand you always have to know where your 5' ends and your 3' ends are. So no matter what, if you're going to replicate DNA, you have to separate the strands. And then you can replicate it. But what do we need to replicate it? Well, we need an enzyme that's going to replicate it. So DNA polymerase is the main type of enzyme that copies DNA. But what's weird is DNA polymerase needs a primer. What that means is it needs something to add. It needs a 3' OH in order to add new nucleotides. So it also needs nucleotides. So we talked about the 4 different nucleotides, and now we can call them DNTPs. And that stands for DATP, DTTP, DCTP, DGTP, but it's easier to say DNTP. So these are the nucleotides. So you need DNA polymerase, you need a primer, you need a template. So that's very useful to sort of keep in mind. So what DNA polymerase needs is it already needs to have something here. So this is going to be our primer. And that primer has an OH, and then DNA polymerase can take nucleotides. Let's say there's an A here and there's a DTTP here. DNA polymerase can take that nucleotide and add it here, and it could form a bond there, so that's a T. But the key thing is that this is always a substrate for DNA polymerase. Template, primer, DNTPs. You need those three things in order for DNA polymerase to hop on here and start making DNA. So that means DNA replication can only happen in one direction. It means if you have a template that's 3' to 5', and you need some kind of primer, and that primer is going to be 5' to 3'. It's going to have a 3' OH, but then all the new DNA has to be added to that 3' OH, and then there'll be, if you had one nucleotide, then now there's a new 3' OH, and now you can keep adding and adding and adding and adding. So remember this for the rest of your life. Template is read, READ, in 3' to 5' direction. But more importantly, new DNA is made or synthesized in the 5' to 3' direction. And that's always, 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 always the case. Always made in the 5' to 3' direction. And if you remember that you need a template and you need a primer, and you always have to make the new thing in the 5' to 3' direction, then you just have to satisfy those rules when you're answering various questions. And the reason for this is, for whatever reason, the DNA polymerase has to add on to an existing 3' OH. If you just give it a template and nucleotides and DNA polymerase, it just can't do it. It just can't start. So adding nucleotides at the beginning without a primer is called de novo synthesis. And DNA polymerase can't do de novo synthesis. So let's make a little room for that. So remember this little phrase. DNA polymerase cannot do de novo synthesis. What does that mean? It means it needs a primer. So that's a rule. So no matter what we do with DNA replication, that's just the rule. Let's do it. Let's say it again. It's really super important. You need a template. And you need a primer. And then you can make new DNA in this direction. Now we got the other strand. That's going to need a primer. And then once you have a primer, you can make the new stuff in this direction. But notice the new stuff's being made 5' to 3' here, and the new stuff's being made 5' to 3' here. The arrow is always the 3' end. So we're going in the 5' to 3' direction. Alright, now buckle up and we will do some DNA replication. And then we'll go through and see what their slides look like, just to sort of reinforce what we talk about here. Alright, so we're going to do theta replication. It means we have a circle, because this is an E. coli. It's double-stranded. So this is your E. coli double-stranded DNA. And DNA replication starts at the beginning, and it's called the origin or the ORI. So the first thing that happens is that there are some proteins called initiator proteins that can recognize the ORI and bind onto it. So there's your initiator proteins. And then DNA helicase will will start to unwind the DNA. So helicase will start to unwind the DNA. So we're sort of unwinding in this direction and we're unwinding in that direction. Now, because the DNA replication is going to work its way around the circle in that direction, it's called bidirectional replication. So it binds at the origin. I mean, you can certainly imagine where it just starts at one spot and goes all the way around, but it starts at one spot and it goes left and goes right, and then at the end it somehow meets at the end, and then at the end you end up with two circles. And exactly how all that gets resolved is a little bit hard to draw out, and so we're going to focus in on what's going on at one of these replication forks. So we got a bubble, and we've got a couple of forks. So let's look, we're going to zoom in on one of these forks and see what's happening. So now we've got a replication fork. We've already unwound it a little bit. So I think we can make this pretty big. So we're zooming in on a fork. So here's a replication fork. Overall, replication is going in this direction because the fork is unwinding in that direction by helicase. So helicase is down here somewhere, unwinding the DNA. And then to sort of keep track of what's happening, we need to know where the 5' and 3' ends of this DNA is. So let's say you can, and it doesn't matter as long as you're consistent. So 3', 5'. So we've unwound the DNA, and now we want to start replicating it. And what do we have? We have DNA polymerase, we have nucleotides, we have all the stuff that we need, but DNA polymerase can't start because, and so this is a fork. This is still going this way, and this is like the origin here, and we're going to do our replication in that direction. So how does DNA polymerase start? It can't. We need a primer. So we need a different enzyme to come in and make this little primer here. And the primer is made out of RNA. And the enzyme that does this is called primase. So primase can make an RNA. Now that's because primase is a different kind of enzyme. So I said that DNA polymerase can't do de novo synthesis, but primase is an example of an RNA polymerase. So that's kind of weird. So we've got an RNA polymerase involved in DNA replication, but its job is just to make this little primer. So we'll make our new DNA green. Once this primer is made, then DNA polymerase can extend the primer in this direction. And notice, so all of the synthesis is happening in the 5' to 3' direction. Now let's pretend that and then, well, I guess we won't pretend, we'll just do it here. And so now here, we can make another primer, but we have to make it going in the other direction. And it can be, but it's still done by primase. And then DNA polymerase can make DNA going in that direction. So we have DNA synthesis going in two different directions, and this is because the DNA is anti-parallel. And so here, this is the template, and here, this is the template, and the template has to be read in the 3' and 5' direction. So if you look at the template, and we look at what direction everything's happening relative to the template, it's going 3 prime to 5 prime, but we hardly ever think about that. We more think about what's going on with the new DNA that's being synthesized, and that's always in the 5 prime to 3 prime direction. And then we got DNA being synthesized in the 5 prime to 3 prime direction this way, relative to the new molecule that's being made. Because look, here's a 5 prime, but that 5 prime is related to the template. Alright, so the enzyme that does the majority of DNA replication is called DNA polymerase 3. And if you look on the course web, there's a list of all the enzymes and what they do. And so here I just want to point out uh, what they do sort of as we go. And we can, we got unlimited number of PowerPoint slides here. So we can start a list of the enzymes. So far, what do we got? We got we had initiator to get things started, and that binds the origin. Helicase unwinds DNA. There's something called gyrase, which is an example of a topoisomerase. And what's going on there is, as the helicase is going this way, this DNA starts to get all tangled up, and it starts to kind of loop and form supercoils. And so that's just where the DNA loops, and then the gyrase reverses that. So it basically cuts the DNA and kind of twists it the opposite way. And so it just kind of keeps, if you kept, this is like sort of if you had a rubber band and you kept twisting it and twisting it and twisting it, it would get all tangled up, and the gyrase sort of relieves that tension. And then the other type of protein that's involved here is single-stranded DNA binding protein. And so that's just, if once you unwind this DNA, it's going to want to snap back together because A's pair with T's and G's pairs with C's. And so the single-stranded DNA binding protein does exactly what it sounds like. It binds single-stranded DNA, and it keeps it from snapping back together and forming double-stranded DNA. So those are the main proteins that are cruising along at the moment. And then primase makes a primer. Makes RNA primer. And then DNA polymerase 3 does the main synthesis. I know it's called 3. It's the first one that works, and it would be better if it was called 1, but it's not. It's called 3. All right, so how far have we gotten so far? We've got our little DNA fork going. And let's say we've been going for a while now. So this original strand that we had a primer up here. Then we had some new DNA. And let's say we had gotten to about that point before. And at that point, so when it gets to this point, so if we're making new DNA, then we have to unwind the fork farther. And then as we go a little bit farther, we have to unwind the fork even farther. So this DNA at the top, as we unwind that fork, it can just keep going. And when you draw these things, these guys are connected. So these are connected because we're adding nucleotides onto the primer, and then we're adding it on, and we just keep adding it and adding it. Now if we go back a couple slides and we say that's where that fork used to be, and we had made that DNA, and we made it going back in this direction, 5 prime 3 prime always. Now when we unwind this fork, we can't add on to this 5 prime end going this way. We can't do that. So we have to wait until we've unwound the fork a little ways, then make another primer, and then extend that. And so we just keep doing that over and over again. And you'll notice, but when it gets up to this back end here, it has to stop. It hits the back end of that RNA and it doesn't know what to do. So that's DNA polymerase 3 again. That's making this DNA. Because this DNA up here can just kind of keep going towards the fork, that's called continuous synthesis. Because this has to go in starts and stops, it's called discontinuous synthesis. Because this, as soon as the fork opens up, that can just keep going. That happens a little bit sooner than the other one, and so that's called the leading strand. Because this one has to start and stop, it has to wait for the fork to open a little bit. This is called the lagging strand. And these fragments that are generated, so this and this, those are Okazaki fragments. So Okazaki is the guy who figured out that there was discontinuous synthesis, and the fragments are named after him. All right. So now we've got to this point where we've done a lot of replication. Kind of back up a little bit, sort of zoom out. This one is continuous, and so it's just continuously going towards the fork. Down here we've got all these little primers that were laid out, and we've extended some of those, but we've got to the back end of that previous primer and we have to stop. Alright, so now we get to this situation, and eventually the whole thing will be like this. So we'll have a circle, and on one strand it's going to be going like this, and then we'll have another circle that's sort of, they're kind of linked together still, but on the other circle let's see. Yeah, so that'll get all the way to the other end. And then on the other circle, we'll have a bunch of these little guys. We'll have little primers going this way, and these will be extended back that way. And actually, on this other strand here, because when we had our original bidirectional synthesis, we got synthesis going that way and synthesis going that, or the forks opening that way and the forks opening that way. On this other one, we've got our lagging strand is on the other half of the molecule. So this is a little bit hard to visualize, but students sometimes like to kind of see what's going on there. This one we don't really need a primer because we can add onto there. And so this one and this other guy, this strand would be the leading strand on this circle. So that's a little bit hard to visualize maybe, but what we really care about is that what we end up with are this situation where we got one strand, one area that's all continuously synthesized, and then we got this other area that has all these Okazaki fragments due to discontinuous synthesis. So now we have to get rid of RNA primers and then connect all the fragments. So the way that works is another enzyme comes in called DNA polymerase 1, and DNA polymerase 1 is going to come in with a little eraser and erase the back end. Oops, Erase too much. So we'll try that again. Um, so it comes in with a little eraser, and it erases the back end of that. So it has to erase it from the back end. So what this enzyme is doing is you've got a template. You got this old primer that was there. You got this new DNA that's coming up this way. So we need to get rid of this DNA or this RNA and replace it with DNA at the same time. So we just got rid of that RNA, and at the same time, the same enzyme replaces it with new DNA. Same thing happens down here. We erase that, and it's got to be erased from the back end. Replace that with new DNA. And so now we've replaced the RNA. So we got rid of the RNA, replaced it with DNA, and then we got to connect these fragments. So those fragments haven't been connected yet. The enzyme that connects those fragments ties it up in a little bow and just makes a bond there. That's called DNA ligase. And that's forming a bond between those two things. So when we've got this situation, and replaced the primer with DNA, and we still have this little gap there. That's called a nick, because only one strand is broken. So this little strand right there is broken. And then DNA ligase makes a phosphodiester bond. So remember, phosphodiester bond. So it's linking two nucleotides together. So it's just sealing that nick. All right, so that's good. Add a couple new slides. All right, so we've done all that. We've just done, we had to have an initiator protein bind, maybe a single-stranded DNA binding protein to keep the strands open. Probably helicase goes in between there because it has to unwind the DNA. Then we have primase that makes a primer. DNA polymerase 3, which is the main DNA synthesis enzyme. DNA polymerase 1, which removes the primer and replaces it with DNA. And then DNA ligase, which seals the nicks and forms phosphodiester bonds between the different fragments. You could also think of it as linking or tying together the Okazaki fragments. Alright, so initiator starts things off. Unwinds DNA, helicase unwinds DNA. Single-stranded binding protein binds single-stranded DNA and kind of keeps the fork open. Primase makes an RNA primer because it's an RNA polymerase and it can do de novo synthesis, whereas DNA polymerase cannot. Makes DNA, removes the primer, and replaces it with DNA. Then DNA ligase seals the nicks, and everything's all fixed up. So a couple of other little tidbits to point out, and then we'll see what the PowerPoint has to say and point out kind of how they're representing the same information. So the first thing is, so we've got our template, and we've got a primer, and now we've got DNA polymerase 3 is adding new DNA. I think before I had my primer being red, but now we're adding our new DNA, and DNA polymerase 3 is just cruising along, and as it cruises along, it keeps making new DNA. So that's how it's made. Now sometimes DNA polymerase makes mistakes. So DNA polymerase is going along, and maybe there's a G here, and it puts in a T instead of a C, and then it keeps going for a little bit. And then, so this is DNA polymerase 3 still. And then it realizes that it made a mistake. So now it actually goes back and fixes it. So it actually will erase, it'll move backwards and erase the DNA that it made until, so it's, I don't know if I'll be able to erase the whole thing, we'll see. So it starts erasing from this end. It erases all of that and then replaces it with new DNA again and fixes it this time. So before where it had a G there, now it puts in a C. So this backspacing, or deleting it going backwards, is a lot like base backspacing, and then retyping to sort of fix the mistake. Together, these things are called proofreading. And so, but the fancy word for this, so there is proofreading. But this erasing part, so after it's made the mistake, and now you want to think about the enzyme as sort of chewing up like a little Pac-Man. It's going to chew it up and erase it, going back in that direction, and then it'll retype it. That erasing activity is going, now it's going backwards. So it was made in the 5' to 3' direction. Now it's erasing in the 3' to 5' direction. Erasing or backspacing. But the official word for that is 3' to 5' exonuclease activity. So proofreading consists of two things: 3' to 5' exonuclease activity, followed by 5' to 3' polymerase activity, which is what synthesizing DNA is called. So together, that is proofreading. All right, so one more thing, and then we can see what they have to say. So here's our template, and I think my primers were red before, so we'll keep them red. And we got a bunch of primers, and then we've got new DNA, and we've come up to this situation where the new DNA has run into the back end of the primer that was synthesized before. Alright, so that's our situation. We've got DNA polymerase 3 has just finished. So DNA polymerase 3 has just made the green stuff, the new DNA. But DNA polymerase 3 can't deal with this, the back end of this primer. So now DNA polymerase 1, well, let's make it purple. So DNA poly 1 comes on here. And what DNA poly 1 can do is it can erase this primer from the back end. So DNA polymerase 1 can, so we got this primer, and so the problem is when this new DNA gets to the back end of that primer, we have to get rid of it. So we have to get rid of it from the back end. And DNA polymerase 1 can put in some new DNA. Let's make the new DNA purple. So now this is replacing that primer with DNA. So this is DNA. And remember, this is RNA. And so DNA polymerase 1 just did that. And what did it do? It erased the primer from the back end. Well, what's the back end? The back end of this primer is the 5' end, and the front end of the primer is the 3' end. So now it's erased the primer in the 5' to 3' direction, and we call the erasing activity exonuclease. But at the same time, it's doing 5' to 3' polymerase, because it's erasing and typing at the same time and in the same direction. So let's just do that one more time. We've got template, primer, another primer down here. I know my colors are a little messed up, but we got new DNA coming in. Gets to the back end of that primer. DNA polymerase 1 gets rid of that from the back end. That process is called 5' to 3' exonuclease activity. So exonuclease means it's chewing it up from the end, and DNA polymerase 3 does 3' to 5' exo, and this is doing 5' to 3' exo and 5' to 3' polymerase at the same time. So it's sort of over typing. So whereas the other one was backspacing and retyping, now we're over typing the primer and replacing RNA with DNA. So we erase that from the back end, and we put in some new DNA. And that's all done by DNA polymerase 1. Alright, so one more new slide. Make a little list. We've got the initiator protein, gets things started, binds the ORI. Helicase unwinds DNA. Always forget about these guys. Single-stranded DNA binding proteins. Keeps the DNA unwound. So we'll say keeps it single-stranded. Primase makes primer, which is RNA. DNA polymerase 3 does main DNA synthesis, but also does proofreading. And proofreading is 3' to 5' exonuclease activity. We'll just say exo, and 5' to 3' polymerase activity. DNA poly 1 It's also a DNA polymerase, but its specific function is to replace the primer. And it does that by doing 5 prime to 3 prime exo and 5 prime to 3 prime polymerase at the same time. So remember what it's doing this. We said all RNA polymerases need primers. And so that's what DNA polymerase 3 did. And now there's a primer. And this is here. Now DNA polymerase 1 can get rid of this primer, but it can also add on to this arrow there because there's a 3 prime OH there. So all a primer means is that we need something that has a pre-existing 3 prime OH to add on to. Alright, so that's basically it. And all those things are listed in the outline on the course web. And let's take a look at this. Oops, dropped my pen. Now let's take a look at what the PowerPoint has to say about all this. Well, first, let's see. Maybe let's back up a little bit. All right, so we did the semi-conservative stuff. Everything I just showed you was theta replication. These diagrams look a little goofy standing by themselves. And alright, so we got all this. Now eukaryotes also do semi-conservative. So the semi-conservative stuff is simply that you start with an old strand, and then when you replicate it, you get an old strand and a new strand in your new DNA molecules. In eukaryotic replication, the chromosomes are a lot longer, so you have several different ories. You have a bunch of different ories. Sometimes they will call an ori in eukaryotic cells an arse. That's the same as ori. And let's see. What happens at these ories is helicase comes in and unwinds the DNA, unwinds this DNA, unwinds this DNA, and then replication happens in both directions until the replication bubbles sort of run into each other, and then eventually you copy everything. But everything that's happening inside of these little replication bubbles is pretty much the same as what's happening in bacterial DNA replication. Some of the enzymes are called different things. Instead of DNA polymerase 3, it might be called DNA polymerase alpha. And so there's more polymerases in eukaryotes, but the fundamentals are the same. You still need to have a template and a primer, 5' to 3' exonuclease, all that kind of stuff is the same. The specific names of some of the enzymes that do it might be a little bit different. Oh, this term replicon. Replicon just means a replication unit. So kind of from here to here, we're going to replicate that in sort of one step. So that would be a replicon. In bacteria, the whole thing is one big replicon because we're just going to replicate the whole thing. So this is what it looks like in eukaryotes. You've got a bunch of replication bubbles, and they're all replicating towards each other, and eventually you end up with a couple of new molecules. And keep in mind that there's 3 billion nucleotides in the human genome, so you need a lot of these origins spread out over all the chromosomes in order to replicate everything. Everything's the same, whether it's it doesn't matter. All DNA replication is the same. You got a template, you need a primer, you need some nucleotides, you need some enzymes, particularly DNA polymerases, called different things and different things, and then you can make new DNA. But this is always, 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 always the starting point of DNA replication. You need to have a template. You need to have a primer. You have to add on to the 3' OH, and you have to make your new DNA in the 5' to 3' direction. And you read the template in the 3' to 5' direction. All the time, always, always, always. So here's a little bit of close-up of what's happening. Here's our DNTP. So there's our sugar, and our base, and our 3 phosphates. So that's a DNTP. And we're going to link that DNTP. When we actually link this, and I showed this a little bit a while ago with DNA structures, we're going to get rid of these 2 phosphates. And those 2 phosphates get lost as what's called inorganic phosphate. And so that's what this is. So in that PPI, sometimes called pyrophosphate. When we come back and talk about DNA sequencing a little bit, that might become important. And then it's this first phosphate is what gets linked in the phosphodiester bond when we're making this. So here's our template. Here's the new DNA. So this part, you might think of this as the primer at this point. So that might be the primer. And then we're going to add new. So here's a 3' OH, and we're going to add a new nucleotide onto that 3' OH. In the process, we create another 3' OH. And so here we're losing these phosphates, and this phosphate's forming a phosphodiester bond. Everything's the same, continuous and discontinuous replication. So if we've got a bubble like this, and that bubble's going that way, and that bubble's going that way, we'll have some continuous synthesis in this direction, and we'll have some discontinuous synthesis in that direction. So continuous and discontinuous means we have leading and lagging strands, and all that's the same in eukaryotes, prokaryotes, doesn't matter. Alright, so what's this is just I'm taking a look at your fork, showing that the forks or the synthesis is going in 2 different directions at the same time because the 2 template strands are anti-parallel, and because you have to make the new stuff in the 5' to 3' direction, you have to synthesize DNA in opposite directions. Now what's confusing about this is that the overall motion of this whole process is actually going in this direction. So the fork is opening in that direction, but the actual, when you get down to what's happening at the actual molecules, the synthesis is going right to left in this picture. So it's kind of weird, and you have to watch some of the videos to kind of get a visualization of what's happening. But really what's happening, so this guy, we got this guy, got a primer and going that way, that's no problem. This other strand kind of loops around, and the synthesis is going in this direction. And so it's kind of hard to see. You get your template kind of loops out, and then the synthesis goes in that direction. So it has to there's some looping that goes on. It's hard to draw. But the main point is that this synthesis is all going left to right, even though within that motion, some stuff's going right to left and some stuff's going left to right. All right. Everything's the same. Okazaki fragments you have to get stuck back together, just like in the other thing. I thought I was going to draw some stuff out there. So The rolling circle model, let's skip that. Um, so this is just showing the three different mechanisms. Um, where to go? So it's just showing the three different mechanisms. It's showing the theta over here. We got theta. We got linear replication. Those are basically the same thing. Rolling circle is just a little weirder. This sort of strand gets peeled off, and then you have synthesis of this, and then eventually you'll get um, copying that. Don't worry about rolling circle. It only happens in some kind of weird viruses and that sort of thing. All right, so this is just a review of what we just did. We've got an initiator. We've got the ORI. We've got the initiator protein. DNA helicase, single-stranded DNA binding protein, a topoisomerase that, as the fork is moving in this direction, this DNA gets all sort of tangled up, and then DNA gyrase kind of unwinds it. It cuts it and sort of unloops it so that it doesn't cause too much stress. So there you go. Here's your single-stranded binding proteins. What do you suppose a single-stranded binding protein does? It binds single-stranded DNA. Okay, so primers are usually 10 to 12 nucleotides long. They're made out of RNA. And the primase is an RNA polymerase. So that seems a little bit weird because we're doing DNA replication. But we've got a template. And RNA polymerases, they do not need a primer. So they can start adding on nucleotides right from the beginning. So they don't need a primer to add on to. So this RNA polymerase can do de novo synthesis. De novo just means new or from scratch. Meaning it doesn't have to add on to something that already exists. It can just read that first nucleotide and start adding complementary nucleotides. There's a whole bunch of stuff going on, showing the forks going in two different directions. We've got here, we got the red stuff going towards the fork right here. So we've got a primer. We've got, since the leading strand is going towards the fork, because in this case the fork is opening in that direction. So down here the leading strand is going towards the fork. The lagging strand has to wait a little bit and then can make a primer and go in the opposite direction. As that fork opens up more, the leading strand just continues to go, but we have to keep making primers on the lagging strand. The same thing happens on the other side, where the leading strand is going towards the fork. So this leading strand is going towards the fork, and the lagging strand is coming back in fits and starts, going the other way. Elongations carried out by DNA polymerase 3. Elongation just means the main DNA synthesis of making the DNA longer, so they call it elongation. DNA, I don't know if they talk about proofreading here yet, but this also does proofreading. And then DNA polymerase 1 removes the RNA primer, and then DNA ligase connects those nicks after things are done. And there's some kind of mysterious termination protein that separates the two new molecules and makes new chromosomes. So this is good to know. This is all we really care about in E. coli. There's a lot of different polymerases, only a handful in E. coli, but so we talked about 1 and we talked about 3. All these are different DNA polymerases, so they have to do 5' to 3' polymerization. That's what makes them a DNA polymerase. And then DNA polymerase 3 does 3' to 5' exonuclease, and that is the proofreading function, but it cannot do this 5' to 3' exonuclease. DNA polymerase 1 can also do this proofreading business, but mainly the thing that's important about it is that it can do 5' to 3' exonuclease. So proofreading is erasing in this direction, and that is 3' to 5' exo, and then 3' to 5' to 3' exo is when we're erasing in this direction from the 5' to the 3' direction and replacing it with DNA at the same time. So DNA polymerase 1 does 3' to 5' exo. Does the other one, but we don't care about that. And this one does 3' to 5' exo. And that's proofreading. So a lot of enzymes do proofreading, but in the context of this, we really want to keep. You'll never get it wrong if you say that DNA polymerase 1 doesn't do proofreading, but the DNA proofreading is more important with DNA polymerase 3. All right, so after we've done all that, we've got this little nick right here. And this is what we mean by a nick. One of the strands hasn't been connected yet. So when we're looking at this, we can see that synthesis was going right to left because the bottom strand is being synthesized in this case, going 5' to 3' direction. When eventually DNA polymerase gets done, it can't make the final connection right here. And so the enzyme that does that, so this is saying DNA and RNA are butting up next to each other. The DNA polymerase can chew up and get rid of the RNA and replace it with DNA, so that's what this is saying. And then at the very end, DNA ligase fixes that little nick. And there's the list of what everything does. This talks about the proofreading. We already talked about proofreading enough. Mismatch repair is kind of interesting. So proofreading happens while DNA is being replicated, but this mismatch repair is sometimes the proofreading misses it, and you'll get some kind of you'll get a mutation, you'll get a mismatch. If it doesn't get repaired, it becomes a mutation. And there's this process called mismatch repair, where there are repair enzymes that can recognize that repair and replace it. There's a lot of details, but we've been going on forever on this thing, so just know that there is some repair that happens during replication called proofreading, and there's some DNA that happens after replication, and one type of that is mismatch repair. Oh boy, which mechanism requires the ability to distinguish between newly synthesized and template strands? So this is mismatch. Well, it should be, hopefully it is. The idea is that because, let's say we've got a mismatch like this, and this was the template, and this was the new DNA. If we're just going to, if we have a mismatch, if we have a G here and a T here, if we want to repair that, we could just kind of randomly, so it should be a GC because the template started out as a G and we were trying to copy it, so we shouldn't put it in a C. But we put it in a T instead. But after replication occurs, the repair enzyme has to come in and try to figure out, it has to fix that and says, all right, well, should I take the T out and put it in a C, or should I take a G out and put it in an A? So there are systems so that the enzyme knows that this was the original strand, and so it'll take out this T and put it in a C. It could just do it randomly, but if it did that, then 50% of the time you'd get a mutation. So all this is saying is that it requires the ability to tell which is the old strand and which is the new strand, so it can replace the mistake that was in the new strand. Whew, I got it right. Okay. Eukaryotic replication is the same. Some of the words are a little bit different. We have autonomously replicating sequences, which is the same as an ORI. We won't worry about the licensing factor that just sort of regulates when DNA replication happens. Eukaryotic DNA polymerase, there's a whole bunch of different eukaryotic DNA polymerases. So there's a humongous table of them. I like this table because it tells you what all those Greek letters are. I can get, you know, up a little ways in Greek letters, but once I get to iota and maybe zeta, I might not be able to recognize those. Maybe you guys would. Maybe it's part of your fraternity thing. So basically, the idea is that there's lots of different polymerases. So let's see. So DNA alpha. So alpha has its own primase activity. So it's a complex enzyme that can do primase and it can also get DNA synthesis started. Your main leading strand synthesizer is epsilon. Probably the one, doesn't really say which one replaces the primer. But the idea is that it's a little more complicated. There's lots of different enzymes, but all the fundamentals are the same. So you don't really need to know all these different enzymes. Just know what happens in E. coli, and know if you ever study eukaryotic replication, you should be able to tell which ones are which. All right, and we talked about the other thing that's important is that because chromosomes in eukaryotes have histones, you have to deal with the nucleosomes. You have to take them off and put them back on. We won't worry about exactly how that happens. We will talk about telomeres. And I'm going to stop this here because the telomeres are going to have their own separate lecture on how telomeres and telomerases work.